that. I have just a screenshot of that coming up. This is contact information for myself and Dr. Cloud. It's sort of a weird course because there's no book. So we can never like get together and say, here's a book. And then as a student, it's like, okay, you don't have a book, and what do you do? And so we don't have a book because we think that, well, there isn't really a good book to teach what this course needs to teach. And so what we do is we try to put together a really complete note set. So really great note set in there, embedded in there, but it also depends on you all keeping up with the material. Now what you don't want to do is say, well, I'm going to use the PowerPoints to study, because the PowerPoints are terrible to study. I have a picture of like people fishing for giant squid, and you're going to be studying, and you're going to have like 20, 90 slide PowerPoints to go through, and there's going to be like clouds, like our first slide is like clouds, and you're going to be like, what's he thinking? So use the note sets for that, and also you'll see that I have summary PowerPoints that really refine what we're doing. And as we go through the class periods, you'll actually start to put those together. So as we have extra time, we'll work in groups and we'll do those, and that'll consolidate the information for you and give you a really nice set of material to work with. So this is our little syllabus. So little syllabus is just when the class meets and what we're going to talk about. And then the big syllabus I consider to be the note sets that are included. So you can use those. I've broken them down. So there's one for every class day. Sometimes one for 2 class periods. I'll note that. And I split it up because last year people didn't really like having one document, having like one 200-page thing, like a book. They didn't like that. They liked having it broken up into what, in effect, are chapters. So if you go through those, you'll be in really great shape. If you go through the summaries, you'll be in great shape. And then class is sort of a time when we can talk about things, so you can ask questions. You can try to stump me, and I'll say, oh, I don't know, I'll look it up. And then the next day I'll come and say, oh, that was really neat, I looked it up. Here's a bunch of papers you can read, enjoy. So my office hours are 8 to 9:30, Monday, Wednesday, Friday. You're probably doing something. You're probably at WSU. By appointment, I'm at work a lot. I will be in the building somewhere. I'll make time for you. Just send me an email, and I'll schedule you in right away. Usually that's what people prefer to do. We found that there's a lot of differences in people's background in this course. So we might have someone, two years ago we had someone who had a PhD in physiology. All right, and so it's like, okay, he was that. And then legitimately, like some of you majored in microbiology, maybe you didn't major in a science, and so you'll have to put in a little more time. And that's going to be true of all of your courses. Some of you will be really strong in neuro, maybe you majored in neuroscience. And some of you, those students are going to have to spend a lot of time in microbiology. And so it all sort of works out. In the first year, basically, we're just trying to get you all to the same place. So if you know this, that's fantastic. That's so great. I would just say make sure to concentrate on the exams. The only time we ever have students who really have trouble is if they know the material really well and they're like, oh, I'm going to like not study and study for something else. Or if there's just some big life issue that they run into. So those are sort of the two things. And if you just put in the time, you will be set. You may have noticed if you look at the syllabus that the course can roughly be broken up into 2 halves. So part 1, I'll cover all of this material. These are our topics through exam 1. So we're going to talk about how cells can detect differences in the environment, basically, and how the organism uses that. So how that underlies vision and hearing and our other senses. And then we're going to have an exam. Pretty typical. The second part, I'll cover 2 of these, and Dr. Cloud will cover the rest. And then this is going to be different from your other courses, but we found it worked very well in the past. It saves you a lot of time. On Thursday, a Thursday of exam week, we're going to have exam 2. So in the morning, Dr. Cloud and I are going to get those exams. We're going to sit down and we're going to grade them right away. Grade them, get them back to you. Then your final is the next day. And so it seems like, oh my gosh, this is crazy. But actually it works very well because the second part of the course really builds on the first part of the course. And it's almost extra work to have to study for the second exam and then have to study for a final. It's just like extra time. And so we found that this works really well. We've never had a problem with this, so if you're concerned, we've actually never, since we started doing this, we've never had a student fail the course. And so that seems to work pretty well. If there is a problem where we grade your second exam, it's like, oh my gosh, then we'll work with you. So we'll make this work. It's something to be happy about, not something like, oh my gosh. Like it gives you a lot of extra time. So when you have, exam bank 1, you'll be overwhelmed. Exam bank 2, you'll think, oh gosh, we don't have self-phys this time. This is awesome. I can focus on my other courses. And then you'll have self-phys again with the other finals. So is that everyone understands that? So you need to do 2 things to pass this course. So you need to get a 70 basically as your average, and you need to pass the common final with a 70. So the common final is common to all sites, so all summer long we shoot emails back and forth and we say, oh, should we have this question, should we have that question? It's going to be computerized this year, and the reason it's going to be computerized is that UW has found that when students go to take their boards, that often taking the computerized test throws them off a little bit. And so actually just having some exam that's computerized actually helps a lot. So it kind of gets rid of that stress, then you're used to doing this. And so we're transitioning all of the courses eventually to be computerized. You're really a special class. So why? So you're the last class that's going to have 1 year here and then go to Seattle. So in the future, it's going to be 18 months, then 6 months of prep for the boards basically and some other things, and then you do your final years. You're our first year of all Idaho students. We're very excited about that. Don't let that get back to Seattle, but we are. It's really great. It's great to have a more workable number of students also, so 30 students. It's really great, and I think the instructors were all pretty excited about that. So when you look at Canvas, you'll notice a number of different things. This is just a screenshot. I will have these worked out completely the Sunday, like by Sunday evening of the week before. You notice there's something up already for all of my courses, but I might not have made a quiz yet or some such, so you don't need to take a quiz before you have the class, so I'm not too worried about it. The quizzes are for you. I'm making them entirely for you. They're entirely for practice. No points are awarded for them. It's what I think you should go through, and as long as you're not missing more than 1 or 2 questions, you're okay. It's just a test of basic knowledge. Did you get it? It gives you an idea of what I think is important in terms of the examination. So in the course, we cover a lot of biology. You'll see today we cover some history of how things were discovered, and we discuss practicality. So the things that are practical are important, and the biology is important. The history is hopefully something to make it more interesting, but beyond that, you don't need to know it. And so that's why I have the quizzes, just to give you an idea of what you should do. And you can take them as many times as you want, and just see it's similar to the question structure that you'll have. Again, we have a part 1 and part 2. And today we're going to talk about homeostasis. That's awesome. So when I was in the 4th grade or 5th grade, I don't remember, I was in science class and we learned cumulus clouds and nimbus clouds and stratus clouds, and they were these like fluffy clouds, and so that was great because then you could name clouds. And we also learned about osmosis and homeostasis. So homeostasis has forever been this word that's been like homeostasis. It's like this word, and you define it, and it's like, well, there's sort of a balance, and you know that's what it's about, homeostasis. So really, this course is going to be about homeostasis. And if you think about it as a doctor, like 80% of what you're going to do is helping people to maintain homeostasis. So you have this normal range that you operate in. You have, if you move out of that, disease, and then if you move too far out of that, you can have mortality. And so what you're going to see is that in understanding physiology, we can use physiology to move someone back into a range of normal homeostasis. And if we think we are multicellular organisms, so we not only have to maintain what's going on in our cells, so if you're a prokaryote, you just have your cell and you signal to other cells, but we are multicellular, so we have to signal to other cells. Your cells are specialized, and you can die. So if you're a bacteria, you can just sort of divide and divide and divide, and you can die, of course, but you could keep dividing, but here we're going to try to avoid death by trying to keep us in that homeostatic range. Now, I want to focus on case studies a little bit. So we have 2 case studies, so we're going to cover these now. We're going to go through the class material, and then we're going to come back and discuss them and see how what we learned can help us. So we have 35 years. That's a very young man, younger than me, was admitted with acute nephritis. So what's nephritis? Inflammation or infection of the kidneys. And this resulted in an elevated serum potassium. So normally serum potassium is 3.5 to 5 milliequivalents, and in this patient there was 7. What is a milliequivalent? Who can answer? Come on, I know this must have been on your MCATs. What's the difference between a millimole and a milliequivalent? In this case, nothing. But a milliequivalent can be different from a millimole because it takes into account the charge of an ion. So if an ion, say calcium, is Ca2+, so you have a higher charge, its milliequivalent won't necessarily match up with its millimolar value. So milliequivalent is basically the relevance of it in a physiological situation, where millimolar is what you could measure in a lab. So for now, you could think of these values as millimolar. So the patient also has lower sodium levels, so 132 versus 136 and 145. And this type of infection, this nephritis that throws off the patient's homeostasis, will actually cause neurological and cardiac complications that can be life-threatening. So if you have a patient with an infectious disease, the physiology could cause the patient life-threatening complications. So we'll come back to this and we'll work through this through the course and see how these deviations, so this doesn't seem like a big difference, but we'll see how it really is in terms of the physiology. Our second case, so we have a 64-year-old female patient. She was admitted to the hospital with congestive heart failure, so atherosclerosis or some such. The physician gave her a prescription for digitalis, which is a cardiac glycoside. Does anyone know what this flower is? Foxglove. So foxglove is the plant that digitalis is derived from. So about half of medicines are actually derived from plants or other animals or plants, and digitalis would be one example. And the digitalis is going to make cardiac function stronger. So it's going to make the heart muscles contract more strongly. Unfortunately, this patient was given an overdose, so too high of a prescription. And so what we need to consider is, why was this medication given? So why would you prescribe a cardiac glycoside to a patient? And what would happen in terms of physiology if you were to give an overdose? So what would you do? So those are our two cases, and we're going to finish up with them. A lot of this course focuses on neurons. So we'll talk about muscles, we'll talk about other systems, but especially in the beginning, we're talking about neurons. And I just want to review this briefly, just to make sure everyone is set with where we are. So our neuron, this is my generic cartoon of a neuron. We have a cell body, which I'll call a soma. We have dendrites, which receive input, and axons, which send out output. There's two types of synapses that we'll deal with. Chemical synapses. These are the synapses that use neurotransmitters that we'll talk a lot about, like acetylcholine. And there's also gap junctions. So gap junctions are direct junctions between the cell, and they can transmit electrical current directly through cells. Like, for example, cardiac muscle is all connected by gap junctions, and that helps it to coordinate contraction. So the term nerve refers to a bundle of axons. So we've got a bundle of axons in the peripheral nervous system, so in the body proper. A fiber would be a bundle of axons in the central nervous system. So you would have your optic nerve, which is going from your retina, and then when it reaches your brain properly, it becomes the optic tract, or the optic fiber. And so that's what I'll be referring to. Afferent would be a receptor sending information to the central nervous system. And efferent, so if you think of effect, will send information from the central nervous system to a muscle or to a gland, for example. So is this, everyone's pretty good with this? All right, so here are our learning goals for today. How do membranes act as barriers? This is going to be a little painful. Bear with me. Hopefully it's review, but maybe it's new material for some people. How do molecules move across membranes? This is really important, really central. I'm sure how do molecules move across membranes? What are some ways? Diffusion. All right, so this is going to be review. You'll be pretty familiar with it. That's good. We have some different equations to describe this. The Morse equation, flux and Fick's first law, and the Nernst equation. We're going to be doing the Nernst equation today. So if you have the calculator function on your computer, that's going to be really helpful. That might be new to some people. The others will be pretty similar. We'll see a number of equations that you learned in physics all of a sudden, like Ohm's law, which is great. It's great to, why did I have to take physics? Well, all right, there we go. We're going to talk about resting membrane potential. Resting membrane potential is really key. So your cells have a charge that's different on the inside than the charge on the outside. And that difference is called the resting membrane potential. And as we'll see, that can change, and that's the basis of physiology, of neurophysiology, is the resting membrane potential. And today we're going to talk about what it is, where it comes from, and what it's going. If anyone's seen that famous Gauguin painting of women in the Polynesian islands, and it's called "Who Are We? Where Did We Come From? Where Are We Going?" and they're grinding corn or something like that and doing other normal chores. It's a great painting, and I always think of that as my idea for these terms. Action potential would be a good one. So osmosis, oh my gosh, osmosis. Isn't this funny? This is like second grade. So osmosis is just transport of water. And in biological systems, it's actually mediated by a protein called a quarin. So chlorine are molecules that help water move across the membrane. Now, osmosis is a pretty old concept that people have been looking at. And one of the early experiments was to take our friend the goat and take out the bladder of the goat, which the goat probably did not like, and fill it with water and put it in water. So there's our goat bladder, and it's in water. And now what our happy monk did was add some salt into the bladder. I don't know if this was a deliberate experiment or not, but what happened? Yeah, so water's going to enter, it's going to move in there, and it's going to dilute out that salt a little bit. So why should we track water? Why is that important? Homeostasis, awesome. All right. You're probably very familiar with this experiment also. Maybe you saw this in 8th grade, maybe high school. You take red blood cells. So you take red blood cells and you put them in an isotonic solution, a hypotonic solution, and a hypertonic solution. So everyone's okay with those terms? Which solution was this one put in? What is it? Hypotonic. So what's happening in a hypotonic solution? Where's the water going to go? It's going to go in the cell, and it's going to boom. And hypertonic, which would mean lots of salt, where's the water going to go? It's going to go out, yeah. So you're going to try to dilute out that salt, and the cell is going to shrivel up. So again, just classic experiment. And there's just a little image of what's going on with water and salt. So based on these, what can you say about the movement of water and ions with respect to red blood cells? So we've got some potential answers here. Has Lee passed out her flashcards to you yet? No? Oh, okay. You're going to get flashcards, and then you hold them up, and that way you're not embarrassed to answer wrongly. But I don't have a problem with that, so I just ask questions. So what's going on with respect to water and blood cells? Which would best describe what's going on?
4.
4. Water crosses the membrane more easily than ions. How do you know that?
Previous experiment.
Previous experiments, yeah, because the water's what's moving more so than the ions. And I used to have this track, so you'd put in answers, but the software never really talks to each other, and so then it was just frustrating. So if I were to summarize this, if you were to summarize this, could I have different volunteers? How would you just summarize the last 10 slides in one statement? John?
Water follows salt.
Water follows salt. Great. Anyone else?
Usually across a semi-permeable membrane, so it's not allowing the salt to move, but it is allowing water to move.
Yeah, so the membrane is semi-permeable. It's differentially permeable, so that's good.
Ions should be very afraid of water.
I heard it, but I didn't see who said it. Who? Josh. All right. So you can see we have 10 slides, and you can summarize it with one statement. And so this is the idea of reviewing this at the end of course and coming up with a list. You'll see some of my 80 PowerPoint slides get summarized to like one slide. And that's awesome, right? You want to study with like 20 slides and not like last year the students tried to do it and they had like 2,000 slides. And they're like, we hate the PowerPoints. All right, so we have actually a formula. So now we're out of like 4th grade. And we have a formula and we hate them. We hate formulas. They're the worst thing ever. And we want to describe the tendency of water to move. And so we use this equation, and the good thing about the equations here, we'll do some in class, but there won't be equations on your exam beyond, well, what happens if we increase this? And this is our equation for osmotic pressure, so the Morse equation. So osmotic pressure of a solution. So what sort of things would influence osmotic pressure? What's it? Molarity. So we've got an M. Temperature.
Number of ions.
It's molarity. All right. So you got most of them. Molarity. So how much of a solute is in the solvent? So how many ions are there? R is the universal gas constant, so that's what it is. So you don't memorize that, look at it, but don't actually make neurons remember that, because when you make memories, you often pound on adjacent cells and you forget things. So making new memories sort of loosens up old memories. And T is temperature, and we're talking about absolute temperature here, which is going to be in degrees Kelvin. And just as a reminder, if you take this number and add your body temperature, for example, you would get degrees Kelvin that we'll use. And basically this makes sense because if you have a higher temperature, the molecules are moving faster. If you have a lower temperature, the molecules are going to move more slowly. I is Van Hoff factor. Van Hoff factor is how much hair gel you put in the morning. And it makes your hair stand up funny. So Van Hoff introduced this factor to take into account different kinds of molecules. So some solutes tend to associate. So if they associate, like alcohols, then they diffuse less. Some solutes dissociate, so sodium chloride. The sodium and the chloride dissociate, and that's going to increase their movement, so salts. All right, so Morse equation, here it is. Which has the greater osmotic pressure and why? I want 3 different answers. On one hand, we have 1 molar sodium chloride. And we have 1 molar sucrose, and there's their molecular weights. Which has a higher osmotic pressure? Nicholas?
Sodium chloride.
Sodium chloride. Who else? That's 1 of 3. Fritz?
They're equal.
They're equal.
That's a sodium chloride.
Jeffrey? Sodium chloride. So if it was sodium, like here, they would be equal. But if it was sodium chloride will be greater because it's a salt and it dissociates. So molecular weight, that's our red herring. It doesn't influence this. So it's independent of molecular weight. Molarity is what to focus on here. So another flashcard. So you have 2 flashcards now. You have how water moves, and you have the Morse equation. And so what we want to understand is how things move across a membrane. So how they move across a membrane in our body so that we can understand physiology, so that we can help to make people better, or at least understand what's going on. And our basic, again, this is, you probably heard this in college, I don't know how many times, is the fluid mosaic model. You have a membrane, and it's fluid, and there's things floating in it, and that's the bimolecular lipid membrane, and it's the fluid mosaic model. When I was a student, this is what I thought a cell was like. I thought it was like a water balloon, and you had a membrane, and you had stuff in it, and there was just kind of stuff in it. And probably that's because I was a microbiology major, and microbes don't really have organized inners, at least compared to eukaryotes. But actually, if you consider a cell, you might think of the membrane more like the wrapping of a skyscraper. So you have all sorts of tubules running around, you have elevators carrying things around. So you have this semi-permeable barrier that allows things to move in or not move in depending on what it is. And that's how you can think about a cell membrane. Again, that should just be review. So in this wonderful cartoon of a beaker, we have a membrane separating two different concentrations of sodium chloride. And what we're going to talk about is flux. So flux is just change. So flux is the likelihood of something to move. And the first thing we're going to talk about is passive flux or passive transport. So passive flux is just movement due to kinetic forces. So we've talked about one thing that will drive a chemical. What's that? Concentration. So what would you expect to happen? Is this sodium going to move here, or is this sodium going to move here? What? What's this one going to do? It's going to move here. What's this one going to do?
Stay.
No. What's this one going to do? It's going to move here, but there's not as much of it. And so you're going to have more sodium moving here than sodium moving here, and it's going to balance out eventually. Eventually you'll have an equal amount of sodium on either side, but these molecules are in motion on either side of this membrane. It's just there aren't very many of them, so there's a low chance that these ones will move over here, whereas there's a lot more over here. I guess small means low concentration, big means, I should have pointed that out. So,
Yeah.
All right, so we've got another equation. So this is our equation for flux. So flux is movement, and we've got a whole bunch of letters, but they're actually really intuitive once you see what they are. So here's our equation. Here's flux, and this is the amount of your solvent that moves over time. The amount of your solute that moves over time. And that's equal to the diffusibility of the substance. So some things are really diffusible. They can cross that membrane really well. Some things aren't very diffusible. And it's negative because it moves along the following gradient. A is the area of the membrane that it flows through. So if you have this little area of membrane, then you're not going to have as much movement. If you have this big area of membrane, you have a lot more space for those ions, for your solute to move through. Dc is your concentration difference. So if you have a small concentration difference, you're not going to have as much movement. If you have a big concentration difference, you'll have more movement because there's more of an imbalance. And then thickness of the membrane, so this is if you ignore biology, if you have a thick membrane, so maybe you'll be implanting membranes into your patients and you'll have to think, well, how thick is this membrane? Is our nutrients going to really be able to diffuse across that? And that's something you'll take into mind. Really all I'm concerned about this is that if I were to tell you, how's area going to affect diffusion, that you know. So I don't even think you have to do much beyond read this. Read it before the exam, you'll be fine. It's just really intuitive, but in the event that you haven't seen it before, there it is. I'm not going to say, what's Flick's first law, or something like that. It just would be silly. So diffusion along a chemical gradient, though, really isn't all that there is. So what we've been talking about is this situation where we've got some beaker and a membrane, and it's in glass, and that's it. And that would be the chemical gradient. But in a biological system, you have to consider the electrochemical gradient. So I told you earlier that there's a difference in the voltage between inside and outside the cell. So ions move differently from inside to outside the cell, and that causes a charge to be built up across the membrane. So now we've got sodium chloride, we've got a salt, and our question is, what's going to happen? So we have our salts, the salts dissociate, we've got a negative pole, a positive pole. What's going to happen? Is this going to happen? Is all the sodium, we know opposites attract, is all the sodium going to get sucked over here to the negative pole, all the chloride to the positive pole? What's going to happen? Are they just going to stay the same?
Be the top right one.
Top right one.
Use electrode.
Why?
You have the electrical gradient, then you also have your chemical gradient.
Good. So you have a combination of gradients. The ions are going to follow their chemical gradient, and they're going to follow their electrical gradient. And over time, you're going to reach an equilibrium. So as you have sodium, for example, attracted to your anode, then you're going to accumulate more sodium here, and it's going to be more likely to move back. Even though it's held there a little bit by the negative charge, you now have more of it, and you'll reach an equilibrium. So you eventually reach an equilibrium, just like you do for just the chemical gradient, and this can be described by something called the Nernst equation. Now the Nernst equation, I'm going to want you to know in a little bit more detail because it's more relevant, it's more important, and you'll see it's actually very elegant. I won't ask you to calculate something on an exam, but I will in class in a second. So we're going to come back to the Nernst equation, but I want to first just review other ways that ions can move. So we had facilitated diffusion, and we can also have active transport. So what happens if you just have a membrane that's not permeable? Your membrane is not permeable to an ion. You've got a bunch of it over here, and you want to move it over there. What do you need? I'm going to hold off on the ion pump.
Channel?
You're going to need some sort of pump, some sort of something, some sort of carrier is what we would use to move that from one side to the other. So think about a membrane. A membrane's lipid, and so maybe it's going to resist moving something that's lipophobic, right? So if something's lipophobic, it's not going to really be able to easily move across that membrane, and you might need some sort of carrier to do it. In this case, we won't use pump because we're not using energy. And so here's just a little cartoon of that. I think in this cartoon it's glucose. You're moving glucose from one side of the membrane to the other. Maybe glucose out of the blood. You've just eaten a meal and you've got a lot of glucose, more than you have in your cell at this point. And the carrier protein can just let it move through. Now this process is said to be saturatable. Saturable. Why?
Because there's only so many channels that they can go through, so if on one side, if it reaches too much of it, then...
Good. So you only have so many carriers, and once all your carriers are in use, you can't move anymore. So this would be the movement, the flux, and you're not engaging all of your pumps, you're putting in more stuff, you're not engaging all of your pumps, but at a certain point, you're going to engage all of the carriers that can move things, and even if you put more of your chemical that you're moving in, you can only move so much. All right, so sometimes passive flux and facilitated diffusion, they're just not enough. So why wouldn't they, what would situations be, what would a primary situation be where you need more? We can't just use a carrier. Yeah, so if you're trying to concentrate something, if you're trying to move something against its gradient, you need to use energy in some form to do it. You can't just expect a carrier to do it because a carrier isn't going to concentrate something against its gradient. And we call this active transport. And you've probably seen this before. We have primary and secondary active transport. Primary active transport, you're concentrating something against its gradient, and you're using ATP as an energy source. So ATP is the energy source. You cleave ATP to ADP, and that gives off some energy, and that energy is enough to overcome the electrochemical imbalance and concentrate that. Now, another type of active transport is secondary active transport. Secondary active transport, you're not using energy, you're not using ATP directly to move something and concentrate it against its gradient. What you're using is another gradient. So you're coupling movement of something in a favorable direction, movement of something in a favorable electrochemical direction, with something against that. And when something moves in a favorable direction, it's a little more favorable, and so it's enough energy to pump something against its gradient. And depending on the direction that these move in, if they both move in the same direction, so one's moving with its gradient into the cell, one's moving against its gradient into the cell, we call that syn port. So syn means with. If one thing is concentrated against its gradient, and what's used to fuel that's moving in the opposite direction, we call that antiport. So syn port and antiport, you'll see that sometimes we'll just refer to them, oh, this is the sodium, calcium antiport pump. And so that's why it's important to know that. We'll actually see that at the end when we talk about our digitalis case. So these are ways that we can move ions, and I thought we're really close to a break. And then we can talk about resting membrane potential. So 10-minute break, everyone can sort of stretch. Last year's class was really funny. They really liked to play hacky sack outside. It was the first time I ever saw people who weren't drinking or smoking pot playing hacky sack. This is pretty funny. But stretch, take a break, and then we'll talk about resting membrane potential.